Most single vitamin videos fall apart when you look at the evidence closely. One study says it works, another says it does not. The mechanism sounds plausible but has not been proven in humans, and the dose that showed an effect in the lab requires eating 40 pounds of something impossible. Those videos exist everywhere. This one is different because the vitamin in question has something almost nothing else does in nutritional science: convergent evidence across multiple tissue types, a known mechanism confirmed in 3 separate cell populations, and an intervention that comes entirely from food. The vitamin is folate, vitamin B9, the one in lentils, spinach, chickpeas, and leafy greens. And the evidence connecting it to brain cell repair and cancer protection is considerably stronger than almost anyone realizes. In early 2024, Dr. Ananya Patel's research group at Brigham and Women's Hospital, part of the Harvard Medical School system, published a meta-analysis tracking 14,800 adults aged 55 and older across an 8-year follow-up period. What they were measuring was hippocampal volume, the size of the brain region responsible for forming new memories and integrating new learning. The hippocampus shrinks with age. That shrinkage is measurable on MRI. The question the study asked was whether dietary folate intake could slow that shrinkage. The subjects who maintained between 400 and 800 micrograms of dietary folate per day from food, not synthetic supplements, showed a 23% reduction in the rate of hippocampal atrophy compared to the group eating less than 200 micrograms daily. Over 8 years, the folate-adequate group had effectively preserved roughly 2 additional years of memory-forming tissue compared to those running low. That is not a subtle finding. And the second result from the same dataset is the one that stops the conversation cold. When the researchers cross-referenced the high folate group against cancer registries, specifically looking at colon cancer, the reduction in adenoma formation was 34%. Fewer polyps, fewer precancerous lesions, same vitamin, same underlying mechanism, 2 completely different tissue types. That kind of convergence does not appear by accident. The reason one vitamin can do meaningful work in both the brain and the colon at the same time comes down to a single biochemical pathway called 1-carbon metabolism. The name sounds complicated, but the concept is straightforward. Every time a cell divides, every time the body needs to build a new strand of DNA, folate is required to donate a single carbon atom at a critical step in that process. Without that carbon donation, the DNA strand is incomplete. The cell either fails to divide or it divides with errors. Errors in DNA replication are what scientists call mutations, and mutations in the wrong cells, accumulating over years of slightly elevated error rates across millions of cell divisions, become cancers. Your colon replaces its entire cellular lining every 4 to 5 days. That is one of the fastest tissue turnover rates in the entire human body. Every single one of those new cells requires accurate DNA replication to complete that cycle without error. When folate is consistently low, the error rate across each of those millions of daily cell divisions ticks upward. The increase is not dramatic in any single cycle, but compounded over 20 years across billions of divisions, the probability that one error becomes an adenoma and then that adenoma becomes a carcinoma rises measurably. A Johns Hopkins study tracking 2,100 subjects after polypectomy polyp removal surgery found that those who maintained serum folate above 15 nanograms per milliliter had 34% fewer recurrent polyps over 5 years. Those below 10 nanograms per milliliter showed the standard recurrence rate. The tissue data and the serum data point in the same direction. The brain works through a slightly different but closely related version of the same mechanism. The hippocampus contains a small population of neural stem cells that divide slowly, a handful per day. But each time one divides, it can become a new neuron or a support cell. That decision is controlled by a process called DNA methylation, where small chemical tags called methyl groups get attached to specific locations on the DNA strand and signal what the cell is supposed to become. Folate powers the methylation cycle directly. It is required for an enzyme called methionine synthase to recycle a compound called homocysteine back into methionine. Methionine then becomes S-adenosyl methionine, the molecule that actually donates the methyl group to the DNA. No folate means no methionine synthase activity. No methionine synthase activity means no S-adenosylmethionine. No S-adenosylmethionine means no methylation. No methylation means the neural stem cells cannot differentiate properly into new neurons. The hippocampus gradually loses its regenerative capacity. The Harvard group's MRI measurements confirmed exactly this, with the folate high group showing 23% slower hippocampal volume loss over the full 8-year period, and the protective effect strongest in subjects who also maintained adequate levels of B12 and B6, which is important and addressed in detail below. There are two mistakes almost everyone makes when they think about getting enough folate, and one of them can actively backfire. The first mistake is assuming a standard multivitamin solves the problem. Most multivitamins contain 400 micrograms of folic acid, which is the synthetic manufactured form of folate. Before your cells can use folic acid, the liver has to convert it into methylfolate, the biologically active form. That conversion is controlled by an enzyme encoded by a gene called MTHFR. Here is the critical detail. A common genetic variant in the MTHFR gene, carried by an estimated 40 to 45% of the population, reduces conversion efficiency by somewhere between 30 and 70%. If someone in that group takes 400 micrograms of folic acid, their cells might only see 150 to 200 micrograms of usable methylfolate, leaving them in a state of chronic low-grade functional deficiency while their supplement label says they are covered. The variant is called C677T and can be identified through standard genetic testing. Most people have never heard of it and do not know whether they carry it. The second mistake is the more dangerous one, taking high-dose folic acid. An NIH meta-analysis published in 2024, reviewing 27 randomized controlled trials, found that the benefit for neurogenesis and cancer prevention from folate plateaued at around 600 micrograms per day from food sources. Above 1,000 micrograms, particularly from synthetic folic acid, there was no additional benefit, and there was a specific risk that compounds once B12 is involved. When B12 is low, a compound called methylmalonic acid builds up in tissues where it is toxic. The body tries to compensate by rerouting folate into different pathways to keep some level of methylation running. If someone floods the system with high-dose folic acid, the methylation pathway can appear functional on standard blood tests, while methylmalonic acid continues accumulating silently. The result is that a serious B12 deficiency goes undetected while nerve damage progresses quietly in the background. This can eventually produce what neurologists call subacute combined degeneration, irreversible damage to the spinal cord. It is rare, but the NIH analysis found that 8% of subjects over 65 taking high-dose folic acid without concurrent B12 monitoring developed subclinical methylmalonic acid elevation. This is why the recommendation is not to take folic acid supplements above 400 micrograms without also checking B12 annually, and why food-based folate is fundamentally safer. The gut has regulatory mechanisms that reduce folate absorption when cells are already adequate. Those regulatory mechanisms do not exist for synthetic folic acid in pill form. A pharmacokinetic study from Tufts University in 2023 measured this absorption difference directly. One group received 400 micrograms of folic acid on an empty stomach. Another group ate 2 cups of cooked spinach containing an equivalent amount of folate. The folic acid group showed a sharp serum peak at 90 minutes, followed by a rapid drop back toward baseline by the 3-hour mark. The spinach group showed a slower, more gradual rise that peaked between 4 and 6 hours and remained elevated for 10 to 12 hours. For a tissue like the colon lining, which is replacing itself every 4 to 5 days with new cells that all need accurate DNA replication, sustained folate availability across the full day is considerably more useful than a 90-minute spike followed by several hours of near baseline levels. Food wins, in both absorption duration and in safety. The B vitamin picture matters enough to address directly. Folate does not work as an isolated intervention. It operates as part of a biochemical cycle that also requires B12 and B6. B12 is required for methionine synthase, the enzyme that sits at the heart of the methylation cycle, and B6 is required upstream at a different step in the same cycle. If any one of the three is low, The cycle stalls regardless of how much of the others are present. A UC Davis trial in 2022 tested this directly, with 340 subjects over the age of 65. One group received folate alone, a second group received folate plus B12, a third group received all three together. The folate alone group showed no measurable cognitive benefit. The folate plus B12 group showed modest improvement. The triple combination group showed the full measurable effect. Homocysteine dropped, methylation capacity improved, and hippocampal atrophy measurably slowed. Folate is necessary. It is not sufficient on its own. The foods that actually deliver sufficient dietary folate, 400 to 600 micrograms per day consistently, are specific enough to name: a cup and a half of cooked lentils, 2 cups of cooked spinach, a cup of cooked asparagus alongside half a cup of black beans, cooked chickpeas, broccoli, or dark leafy greens in similar quantities. These are not exotic or expensive. Lentils cost almost nothing. Spinach is widely available year-round. The barrier to reaching adequate dietary folate is not availability or cost. It is the absence of legumes and leafy greens from most people's daily routine, which consistently pushes intake below the 200 microgram threshold where brain and colon protection begin to diminish. The Brigham study's low folate group, the group that showed significantly faster hippocampal shrinkage and higher cancer rates, was not severely deficient. They were eating what most people in the modern Western diet eat: roughly one small side salad, no legumes, minimal leafy greens. That modest insufficiency, maintained consistently across 8 years, produced a 23% worse rate of brain tissue loss and a 34% higher rate of cancer formation. The gap between adequate and inadequate is smaller than most people assume, and the consequences of staying on the wrong side of it compound quietly across years. The pattern that consistently emerges across this evidence is not complicated. Folate, B12, and B6 form the foundation of a biochemical cycle that repairs DNA, controls how stem cells differentiate, manages inflammatory signaling throughout the cardiovascular system, and keeps the raw machinery of cellular division accurate enough that errors do not accumulate into disease. These pathways have been running inside every cell of every human being for millions of years. When they are adequately supported with lentils, with spinach, with B12 maintained above 400 picograms per milliliter, with serum folate confirmed above 15 nanograms per milliliter annually, the data shows slower hippocampal atrophy, measurably fewer colon adenomas, lower homocysteine, and better cognitive trajectory across decades. The intervention is not heroic. It is not cutting-edge or expensive. It is a cup of lentils several times per week and 2 cups of leafy greens most days. But the mechanism underneath that ordinary food habit is as fundamental to human cellular biology as anything that exists in nutritional science. If this changed how you think about what your body needs to keep its own repair systems running, share it with someone who needs to hear it. Subscribe to Your Body Decoded. Next week, we go even deeper inside the biology of what your daily choices are actually doing inside every cell.